45 minutes. So this is part 2 of our snow day lecture, longer than our normal class meeting because our last snow day, last Thursday, I made a recording that was shorter than our class meeting. And we're just going to jump back into it. And there's our RAS pathway, okay? And we're now jumping into just the heart of Chapter 17 and cranking through these different examples based on this graphic that you should create. And so we're talking about RAS. We learned a lot about RAS already. We're talking more about RAS. And so there's RAS, and it could be normal or it could be mutated. And then here's a receptor tyrosine kinase, and we're going to look at this example now about ERB-B. And ERB-B is also called, when it's normal, it's called the EGF receptor. And so this will be EGF. And that's normal, but this could get mutated. And it could get mutated in our bodies. And it was originally discovered as in an oncogenic retrovirus, where it accidentally got picked up in an oncogenic retrovirus. And so it's been studied since the 1970s, right? So that's like 50 years. And so people know a lot about it. And it's still called ERB-B a lot just because it got so famous. But it is, a mutated form of the EGF receptor. Okay, so we're going to talk about it in the context of this pathway. And you see there's a linear pathway here, like dominoes falling. EGF binds the EGF receptor. It activates the receptor tyrosine autophosphorylation, gets phosphorylated, binds this adapter protein, activates RAS. RAS exchange GDP for GTP. It's active and activates RAF. RAF phosphorylates MEC, phosphorylates ERK, phosphorylates ELK, or maybe MIC, depending on the setting. So we go to ERK, ELK or MIC, and there's some other stories in this too as well. But that's our pathway, okay? And it's important that it's a linear pathway, one step after another, okay? So just a little side note, it's a remembering that that tamoxifen, different story, but just the concept that this drug, pharmaceutical used in chemotherapy of breast cancer, that it works on breast cancers that are ER positive, but not ER negative, because tamoxifen binds, it's an estrogen antagonist. It means it binds directly to the receptor. And ER positive breast cancers have the receptor, ER negative don't. Okay, so it's the same concept. Okay, now we're going to move that concept, just that like logic, that common sense, fact-based concept to this. Okay, so here's just a kind of graphic to show that here's the EGF receptor or B receptor like that, and it could get mutated. And this one is just weird, where if it just loses this part, it is constantly activated. And that happens in that oncogenic retrovirus. That's how it was first discovered, where it picks up a piece of the coding region for the ERB EGF receptor gene. And that retrovirus puts that in the host cell, and it just is active. And that means that RAS is active, and that RAF, MEC, ERK, ALK is active all the time. Doesn't need EGF, okay? So that can happen in the oncogenic retrovirus, or nothing to do with the retrovirus, you could get a mutation. I could get a mutation in this gene, in the coding region, that produces what's called a truncated, a shortened version that is oncogenic, cancer-causing, okay? So don't need to look at this in detail. Scientists at companies have developed antibodies that will stick to the ERB-B receptor and block it, whether it's the normal receptor or the truncated receptor. These antibodies will bind to it and block it. So these antibodies are a chemotherapy. One of them is called cetuximab, and that's the one we'll learn about. Okay, there's RAS, MAF, Merck, like that, right? You know, MIC, Phos-Jun we'll learn about later in chapter 20. Doesn't have ALK on there, that's fine. Another signaling pathway, PI3 kinase, AKT, also player, right? So we look at these pathways one at a time. So there's an antibody that was developed that can stick here, and the companies want to use that to help people who have cancer caused by mutation in this gene in a truncated form that's out of control. Okay, so cetuximab, Wikipedia page. So let's see if I have this. Maybe I don't have it highlighted, but let's just read through it. Cetuximab is an epidermal growth factor receptor inhibited used for the treatment of metastatic colorectal cancer, metastatic non-small cell lung carcinoma, and head and neck cancer. Some specific cancers, right? Why? Those cancers, because EGF receptor is normally regulating the growth of those tissues, and if it gets truncated, mutated, it goes out of control. So cetuximab is an antibody that's given by infusion and made by drug companies. Okay, so now check this out. This is the paragraph that's just the memorable paragraph. In July of 2009, so that's 14 years ago, the FDA approved cetuximab for the treatment of colon cancer, and then here's a key thing for us, with wild-type RAS. Okay, so that's a key concept. They said you can use your new drug to treat cancer, but only if the patient has a wild-type RAS. So let's just go to this figure. Where's that at? So they said, if you have this mutation, so it's growing out of control, you can use your cetuximab antibody to stop it. But only if it has wild-type RAS, because if the patient has mutated RAS, then this pathway is already overstimulated. And it won't help, or it won't help very much, if we slow things down up here. And it's that same concept as tamoxifen. It's like a common sense concept of cause and effect physical relationships. If the problem is here, or if there is a problem here with RAS mutations going out of control, it just is not going to help that patient to slow things down upstream, because the things are going out of control downstream. And that needs to just make sense. You just need to let that sink in and have that till that makes sense, because that's what they're saying. And the FDA says, you got this product, you can sell it, but you can't sell it to people who have mutated RAS, because it just won't help them, because it's active upstream of that problem. Okay, so I'm just going to erase this so you can see that. So, and this is just 14 years ago, and they're like, yeah, this is kind of complicated, but you can use it, and approved it for this. We've seen, it's used now in treatment of these others, but first step with wild type RAS, and not just any RAS, but KRAS. But again, we don't need to learn the H and K versions, the different paralogs. And then they say, since it had little or no effect on colorectal tumors harboring KRAS mutations. Okay? Right? And we just need to be able to explain that logic on an exam, because that's a real clear logic that is used now in this molecular medicine and these, molecular pharmaceuticals for cancer. This is the other reason I look at this. It says, this was the first genetic test to guide treatment of cancer. So in this part of this book, it's all about molecular medicine and molecular pharmaceuticals, focused on treating specific cancer mutations like tamoxifen. Okay, and here it says this is the first genetic test, right? So not the first treatment. There were a couple other treatments like tamoxifen available already, but see what the FDA said in approving setrusumab. They said you need to do a genetic test. So and then it says in July of 2012, the FDA approved a real-time PCR companion diagnostic for KRAS, the TheraScreen KRAS test. So this stuff is, in the last 10, 12 years that this stuff emerged. And there's more of it all the time. And in our course, we shouldn't think of this as commonplace. This is new stuff, and it's really significant. There are breakthroughs, and the breakthroughs are focused on specific cancer problems with specific mutations. Somebody's got this EGF mutation, truncating mutation that's creating this ERB-like out-of-control oncogene. Well, you could treat that with this setrusumab. It could help, but only if the patient has wild-type RAS. Okay, so really, really significant. And this is just Wikipedia talking through it. So, this is now common knowledge. And, if you had a family member, friend, or yourself who had colorectal cancer, a certain type of cancer, you might be reading this to try to understand, what the doctor is suggesting will and won't work. And the doctor needs to understand this, but patients need to be able to absorb this as well to really participate in their medical treatment. Okay, great. So an example from this, right? So we learn about the basics and we dive into it a little bit. Okay, and so, and I don't know why I pulled this in here, but just to say this, so this is just a little bit more about RAS. It says, RAS had been studied here for, must be 50, I don't know why they're saying anniversary for RAS, 2006. I don't know what that anniversary is, but let's just read this. In 1981, right, so, 40 years ago, Bob Weinberg at MIT, working with Michael Wigler out on Long Island, and then Jeff Cooper, our author, at BU at that time. Now, and then a professor at BU, right? He was in Wisconsin. He went out to Harvard. Then he got a job at BU. So he's been working in Boston. And they discovered that small fragments of DNA taken from human cancer cells could cause cancer in mice. And then within a year, those same groups found that the DNA for bladder cancer included the cellular version of RAS, an oncogene that had been found, blah, 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 right? So they're saying, right, in Wisconsin, Cooper in the 1970s, and others elsewhere too, found that RAS was picked up by an oncogenic retrovirus, and then he went to Boston and made this transition, leaving the retroviruses behind and starting to study the DNA within real human cancers, and found that you could find genes that were mutating those cancers that could transfer that cancer to a mouse. And we talked about that earlier in the semester, but a lot of this stuff we want to loop back on because we really want to really be solid on, again, not the names or the exact history, but just the reality of the flow, the timing of this, right? It began back in the 1970s, kind of a long time ago, and then it, just kept stepping up like that, okay? And so, there's our RASes like that, and these are these retroviral oncogenes. There's our ERB-B, there's our MYC, and then, there are others, but we have that short list of 5 that we're learning about. There's SARC, a little bit of that one, right? Okay, so RAS, yay, RAS. And the woman who has the cancer has overstimulation of the PDGF receptor, PDGF signaling pathway. RAS is active under a wound normally, and she just has a mutation where she's making PDGF and activating her normal pathway that way. Okay, here's just more information about her and the chromosomal location, translocation. It's between 17 and 22, and that's that. So just going back to that and this thing. So ERB-B and PDGF and the insulin receptor like that, filling those in. Okay, so then the next thing is when we go to our figure here, we say, yeah, RAS, yay, RAS. Lots of knowledge we have about that pathway, that receptor, and that receptor category. Now we're just going to look at this one, that FOXO works by a receptor tyrosine kinase, so we're at this part of our big drawing, but a different signal transduction pathway that involves activating AKT, and our book explains it, and there it is. So we looked at this briefly before in the first Moody. We get this phosphorylated, autophosphorylated form of the receptor tyrosine kinase activates this PI3 kinase, and this kinase doesn't phosphorylate a target protein, it phosphorylates this transmembrane phospholipid to form PIP3, and PIP3 binds to AKT and PDK, and to collectively activate AKT and release it, an active form of it, the phosphorylated form. That's what's going to phosphorylate it, FOXO, and keep it out of the nucleus. Okay? So here's our third one that's no longer in our textbook. And so we just briefly talk about that. That's AKT. That could be on our exam because that's a famous kinase that regulates FOXO. So just, in this first category, yeah, there's RAS, blah, 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 et cetera, et cetera, et cetera. Lots about RAS. Then a little bit about AKT and FOXO. And now a little bit about this one that isn't even in our textbook anymore, but it's another one that I want us to learn about. And so here's what happens here. It's a receptor tyrosine kinase. It gets activated by binding its ligand, autophosphorylates, and it binds to a protein called phospholipase C. Lipase C. And so it's an enzyme, and this enzyme cuts PIP2. We just talked about FOXO and AKT getting regulated by the kinase. And this is a different protein. This protein cuts it into two pieces. And this PIP2 gets cut into two pieces. And the two pieces are called DAG, which is a molecule that's the tails of the phospholipid and stays in the membrane and binds to and activates a protein called protein kinase C. And the phospholipase cuts the PIP2 into DAG and into something called IP3. And IP3 is soluble and floats into the cell and binds to receptors on internal membranes and leads to the release of calcium. Why are we looking at this? Because it's another profoundly significant signaling pathway. It's different than the first two. It's another receptor tyrosine kinase signaling pathway, and it leads to some small molecules. And in IntroBio, you really likely learned that in signaling pathways in the cytoplasm of target cells, that signal transduction pathways lead to the regulation of some molecules called secondary messengers. And they're called secondary messengers because they're inside the target cell and they're not the first messenger. The first messenger was that ligand, the primary messenger, like that's bringing the signal to the cell, bringing the message to the cell, and it really typically stays outside of the cell. And so we're regulating these secondary messengers, and DIG, IP3, and calcium are three very famous second messengers, like that. And so that's what we want to learn, and it's really just on this figure. And again, it's not in your textbook, so we just need to learn what I'm explaining here. And what I'm explaining here is these guys are in signal transduction pathways, and we just need to be able to describe them as second messengers that get activated by this pathway. Okay? So just to talk a little bit more about this idea that this phospholipid has come up twice. And it can either be phosphorylated or it can be cleaved, like that. So those are our two examples. And We're used to thinking about lipids as being energy storage molecules like fats. They're also the phospholipids of cell membranes. They have other roles like that too. And then this is one that's new, is that they're important in signaling. Lipids are important in signaling. So here we have this phospholipid, phosphatidylinositol, and it's just a member, one of these phospholipids with a head and tails that's in every lipid bilayer. And we don't need to know the structure, but here's the name and structure of phosphatidylinositol. And it can get phosphorylated here on 3 different groups, or it can get cut here and create the DAG that stays in the membrane and activates the protein kinase C, or it can float away and bind to internal receptors that release calcium and increase the concentration of calcium in the cytoplasm. Okay? And it's called PIP2 or PIP3, IP3 for the cut piece. Inositol triphosphate is the cut piece. And lots are known about these molecules because they're very famous. The DAG, just to show you, just remember the name DAG. It stands for diacylglycerol, and that's its structure. It's like the tails that are just left in the cell membrane. Kind of fantastic and interesting. And so there's that image again. Unfortunately, it's no longer in our book. It got dropped from this edition, but that's what we want to learn about that. Okay, and just one more piece, and that is that this calcium is a signaling molecule inside the cell, in the cytoplasm. And it's a second messenger, and calcium is almost always bound to a protein called calmodulin, and then calmodulin is really the signaling molecule. Because calcium, right, it's just this little ion. What's it going to do? But it can bind to calcium and regulate, can bind to calmodulin, and then calmodulin can bind to targets and activate them. So we want to just learn the name of that protein, calmodulin, as part of the calcium signal transduction story and the role of calcium as a second messenger. Great. There's calcium. Calcium does a lot of different stuff. Cell. Like that. Okay? So I think we're getting pretty good on receptor tyrosine kinases. And yes, because we're now going to move on to G protein coupled receptors. And we're not going to talk about them as much. And then we're going to talk about these even less. So we're doing pretty well. And we'll get through some more of this stuff today, and then lots more on Thursday when we'll have class in person and be able to answer questions. So we're going to have a couple examples of this. There are hundreds and hundreds of G protein coupled receptors in our body. But the receptor for epinephrine, or you might know it as adrenaline, is a member of the G protein coupled receptors. They're called adrenergic receptors. So we'll learn a little bit about them. And then dopamine receptor, you've probably heard of dopamine as a neurotransmitter. It also plays other roles in our nervous system and binds to the dopamine receptor, which is also a G protein coupled receptors. And there are many, many examples. And our book talks about some other examples. For example, the receptors for our sense of smell. So when we smell cinnamon, it's because molecules in the cinnamon vapor in the air are binding to protein receptors in our nose that are our olfactory, our sense of smell neurons. And the olfactory receptors are all G protein coupled receptors. Similarly, our eyes, our vision is based on G protein coupled receptors in our eyes. Not that similar, but also. So here's the structure of a G protein coupled receptor. They're all in the cell membrane, and it was pretty easy to find them because when scientists would sequence the coding region of genes, they would see evidence of some stretch of about 20 very hydrophobic amino acids. And that's called the transmembrane region. So it's hydrophobic because it goes through the phospholipid bilayer of the cell membrane. And G protein coupled receptors are easy to spot because they have 7 of those stretches of hydrophobic amino acids in their coding region. And so even before you study the protein, you could just study the genes and the genome and say, look at all those. Those are all G protein coupled receptors. For our sense of smell alone, we have hundreds of, these are called olfactory receptor genes. It's interesting just to note that humans don't really have a great sense of smell. Dogs have hundreds and they have a better sense of smell. And that's because many of human genes are still, the olfactory receptor, G protein coupled receptor genes are still there, but about half of them have gotten mutations, so they're not useful. And so it says of the 636 in humans of these smell genes, G protein coupled receptor, olfactory receptor genes, about half of them are functional, and about half of them are no longer functional. They've got mutations. They're called pseudogenes. And that's because humans don't emphasize our sense of smell, and primates, our ancestral primates, they don't emphasize sense of smell as much as they emphasize vision. And so we have a pretty good sense of vision and not such a great sense of smell, and that's just a process of natural selection. And other mammals have really good senses of smell, and our ancestral mammals were understood to be kind of nocturnal, tree-living, rat-like little mammals that had really good senses of smell because they were active at night. So our ancestors had hundreds of active genes, and those are slowly being lost because we're not using them. And that's called a pseudogene. You can still find the gene. It still has the sequences that allow you to find it, but then you can also find, has picked up mutations that make it defective. And pseudogenes just have to just randomly kind of slowly disappear as we pick up mutations. So just a term and concept in the setting of G protein coupled receptors. So here's the book showing a figure of a G protein coupled receptor, and here's why it's called a G protein coupled receptor, because here's the receptor, and when it binds its ligand, it gets activated, it binds a G protein. So it's a G protein coupled receptor. This is not RAS. It's a different G protein. The best way to remember that is, people don't usually call RAS this, but RAS is a monomeric G protein, just one protein. G protein coupled receptors actually have 3 different proteins forming the G protein. And only one of those is the protein gets activated or inactivated. And it's called a G protein because they have GTP bound when they're active, and then it gets hydrolyzed when they get inactive. So they're similar to RAS, but we're going to be really careful to remember that RAS work on receptor tyrosine kinase pathways, and these G proteins are called trimeric G proteins, and they work on G protein coupled pathways. Really different. These are not related to RAS at all. Not similar sequence, not similar ancestral origin. They both happen to bind GTP when they're active. And so here's just one of them, and it activates an enzyme called adenylyl cyclase, and adenylyl cyclase catalyzes the synthesis of cyclic AMP, and cyclic AMP is formed from ATP, and cyclic AMP is an example of another second messenger. And our book talks about this in some detail, so I won't talk about it very much. But there's one example of a pathway. This is just a figure that shows the activation and inactivation of the G protein. And again, it's the trimeric G protein that gets activated. The inactive form binds GTP. Then you have an exchange. The GDP is released, and a GTP binds. And the GTP key has the protein is activated. The protein is active until GTP hydrolysis occurs. And there's another helper protein that helps it get its job done, but the regulation of it really is still in the catalytic domain, especially of the G protein. Okay? And so this is out of place, but that's okay. Just a summary, it's just out of place. I don't know how it got here. Then here's another one, and another G protein coupled receptor pathway, and this is one that fits with the dopamine receptor. So again, you probably have heard of dopamine. It's a neurotransmitter, and it does different things. It has some different properties. This is just one property it has. Here's the dopamine receptor. This could be dopamine. And we want to learn a little bit about this. So we're going to bind, the ligand's going to bind, it's going to activate the receptor, the receptor is going to activate G protein. The G protein will have GTP bound, and it's going to bind, it's going to activate adenylyl cyclase. We're going to get synthesis of this cyclic AMP. Great. That's a second messenger, so that's going to be floating around in the cytoplasm. That'll be present anytime there's dopamine binding to the receptor. And this this cyclic AMP does a variety of things. One thing it does is it binds to a protein called protein kinase A, and when protein kinase A has been bound, it releases the catalytic domain, or catalytic subunit, I should say. It's not a domain because it's different colors, they're really different proteins, and we have this regulatory protein subunit and this catalytic subunit. The catalytic subunit is released, and look what it does. It phosphorylates a transcription factor, yay. So part of what dopamine can do to a cell is can activate transcription. It can activate transcription through this pathway that we're learning about, and leading to the phosphorylation of this protein called CRAB. And CRAB has kind of a memorable and satisfying name. What happened is scientists were studying dopamine historically, and they found, wow, when dopamine is used on a cell, a lot of stuff happens, including an increase in cyclic AMP, and including increase of expression of certain response genes. And so they did those experiments like we did in chapter 9 to map the enhancer for these genes. These genes get activated, the transcription goes up, they must have an enhancer with the transcription bound to it, factor bound to it. So they did deletion studies to bind this, they did nuclease footprinting studies, they did gel mobility shift assays, and then they purified this protein using something like affinity chromatography. And there are other methods that scientists use as well, like that, but we can think of their discovery in this way. And so when they were doing that and they discovered this enhancer, they said, you know, we should call this enhancer a cyclic AMP response element. Okay? Because when cyclic AMP goes up, these genes, their expression increases. And when they purified this protein, you know, they said what we're looking for is a transcription factor that we should call the cyclic AMP response element binding protein. So that's a satisfying name because it says what it is, and we could just remember the name CRAB. And CRAB is involved in cyclic AMP response, like that, and that cyclic AMP is downstream of G protein coupled receptors, including some G protein coupled receptors that bind to that dopamine binds to. Okay, so here we are here, and we just told this story. So this is an important story for us. We're in G protein coupled receptors. Let's take a look at that story. And that's the story we learned about for dopamine and the dopamine receptor. And again, there are some other receptor stories, different receptors literally, that dopamine binds to as well. But this is one for us. And CREB is a protein that gets phosphorylated, and it's activated, and it's a transcription factor, and it activates target gene expression. So we have a second one of these we want to learn about as well, and hopefully I have my, so this, this is chapter 17. Oh, look, it's a second messenger, like that. And we can play a role here. Cyclic MP does other jobs inside the cell. This is one for us to learn. So, in learning about CREB, and this is actually a list that I used last year, so it gets it's a little bit different, but it's really similar, and I'll update it. But I just remembered that I wanted to update it before I started talking, and I just grabbed it from a lecture from last year. So don't worry about that. Just realize that it gets a transcription factor that gets activated by a kinase. Oh, we have examples of that. FOXO, ALK, we'll get to these SMADs, and then CREB, like that, okay? So, good. Then, here's the other example, and it's going to be epinephrine, adrenaline. Very familiar with that. It binds, as adrenaline or epinephrine is what it's called nowadays. It binds to a receptor on our cells called adrenergic receptors. Adrenergic receptors. And they are G protein coupled receptors. And they are going to cause a response. And it's going to be a really fast response. And I think we actually talked about this in class. I think Ellie asked about it because we looked at this briefly. And that is, here's epinephrine. It binds to an adrenergic receptor, activates G protein. That binds to adenylyl cyclase. We get synthesis of the second messenger, cyclic AMP. Cyclic AMP binds to protein kinase A, to the regulatory subunits of protein kinase A relating to the catalytic domain, right? And in some cases, that catalytic domain phosphorylates CREB. But in this case, we're going to have they're going to phosphorylate another kinase, which activates an enzyme that breaks down glycogen. And glycogen is along polymers of glucose, and so it's a way of storing glucose. And in the cell, like our muscle cells, we can break it down and release inside the muscle cell lots of sugar, lots of glucose, lots of energy. And that's what we need when we have epinephrine. We need to get agitated. We get ready to jump up and run away from the tiger. And so that's our other G protein coupled receptor pathway that we want to get particularly familiar with. And that's a nice one because in this case, there's no transcription factor. We talked about CREB over here. This one, it doesn't lead to transcription. It leads to an enzyme that's going to be a metabolic enzyme that releases glucose from glycogen within the cells, like within muscle cells. And so we don't have to have the sugar come through our blood. The sugar is already there in the cell. It's just trapped in glycogen, and we just release a ton of it. And the muscle's like, yay, I have lots of quick energy to run. Okay? So I think that is, we're doing great because that is the core of what I want to talk about for G protein coupled receptors. And the book talks about them a lot. And we don't need to learn them as well. We don't need to learn them nearly as well as we learn about receptor tyrosine kinases. And it's mostly because of the role of receptor tyrosine kinases so often in cancer. Okay? So that's what's going on. Now, we're going to learn about just a few more receptors that just play unique roles, but really important roles. And so here is an example of, we learned about, this is not a receptor tyrosine kinase. Not in that category, it's a different category. And this category is called a non-receptor tyrosine kinase. Because here, there's going to be a kinase involved, and the kinase is called JAK. And it's not a receptor, so it's a non-receptor tyrosine kinase, and it's JAK. It's bound to a receptor, but it's not the receptor. And when this receptor gets activated, JAK gets activated, and it's a kinase, and it phosphorylates STATs. And STATs are transcription factors. Yay. So we add STAT here. So we like to talk about JAK-STAT because it's really important. Here it's actually a role in the immune system response in being activated. And so we'll talk about STAT. And this is another figure that is a little bit out of date. So I just want to go back and find the better figure that I have of this, and I just don't have it. I was going to talk about these in parts, but here's my big summary figure. And so let's just take a look at this. Nuclear receptors, our estrogen receptors are favorite. Then, wow, we know a lot about these, and we just haven't talked about the KIT receptor, but other ones that we've really talked about now, and we'll keep using them and talk about them. Then G protein coupled receptors. We've got the adrenergic receptors that are going to activate the breakdown of glycogen and that quick sugar rush that happens right inside the cell. And then a different one is one of the types of dopamine receptor that's going to activate CREB, phosphorylate CREB, the cyclic AMP response element binding protein. So we want to know those pathways, and now we moved into this, and we want to talk about JAK-STAT, and we just did. That's as simple as it. And we're going to go through these three other ones. And then that's really all I want to talk about in Chapter 17. But that's just going through this and providing that framework. And then it'll be up to you to help, bring in questions about, well, what do we need to know about this? Because our book really covers a lot of territory. So you could make a numbering system, right? 1, category, nuclear receptors. 2, category, receptor tyrosine kinases. And we'll know most about these. We'll continue to learn more about this. 3, really important, really common, but we just aren't going to emphasize them that much. Then 4, 5, 6, 7. And each of these is a section and a figure in the book near the end of Chapter 17, and they're just like, hey, check this out. Look how these receptors activate a signal transduction pathway, and in each case, it's leading to transcription. So this first one, we activate. Well, we can even back it up here. So for this one, we activate CREB by phosphorylation. Yay. We can even jump up here. For this one, we activate things like ALK by phosphorylation. We also activate other transcription factors like MYC, different story, but we activate some by phosphorylation. Here, we activate by phosphorylation. Here, we activate STATs by phosphorylation. Down here, we're going to activate a protein called beta-catenin by stabilizing it, by preventing it from being degraded. Here we're going to activate a protein called the Notch intercellular domain. We'll talk more about it. And we're going to activate a transcription factor by cutting the receptor and releasing the transcription factor so it can float to the nucleus. And then down here we're going to finally talk about activating SMADs by, again, phosphorylation. Okay, so we're doing great, because we just have a minute left. So we've done it. We've really talked through Chapter 17. We just need to look at these final categories, the figure, just to see a little bit about how they are regulated. And I know this is a hurried look through Chapter 17, but I want to do it. I want to do it today. That's why I tried to take some extra time. And so we'll just look at I might not even have these other figures ready yet. I don't. So I guess I don't. Yep, I don't have them. So we'll have to go back to those on Thursday. But again, it'll be Wnt, Notch and SMADs and the TGF receptor signaling pathways. Yep. So we're in good shape because we can start reviewing this stuff. See, there's the timer. So we're done. So thank you for taking the time, and this will help guide you through Chapter 17, and you'll have lots of questions in class on Thursday, and we'll talk more and loop back over all this stuff. And then next Tuesday, we'll see Riley and get ready for our exam a week from Thursday. All right, super. Thanks a lot, everybody. Take care.